The overall goal of isolating lipid droplets for quantitative mass spectrometry is to understand the function of these organelles, for example in the replication of pathogens such as the Hepatitis C virus.
This method can answer key questions in the infectious disease field, such as why pathogens target lipid droplets for replication.
The isolation of lipid droplets can be done in 6 to 7 hours. During this procedure, it's important to avoid a contamination with keratin.
To begin, remove 50 milliliters from each bottle of DMEM medium and add 50 milliliters of dialyzed fetal calf serum. Next, dissolve 50 milligrams of carbon-13 labeled lysine and 50 milligrams of carbon-13 labeled arginine in 1 milliliter of medium. Mix thoroughly and add the amino acids to the DMEM plus FCS medium. Then add penstrep and glutamine substitute. Sterile filter the medium using a 0.45 micron filter and label the bottle as heavy silac medium. To prepare the light medium, repeat these steps using 50 milligrams of arginine and 50 milligrams of lysine. Label the bottle as light silac medium. Trypsinize the cells and split into 2 wells of a 6-well culture plate. One well should contain 2 milliliters of the heavy silac medium and the other 2 milliliters of the light silac medium. Culture the cells for at least 6 passages. After 6 passages, the incorporation of the heavy amino acids should be more than 95%.